Sample question paper Biotechnology code 045 class 12 2024-25 maximum marks 70 time allowed 3 hours. Friends, you can pause the video and read the general instructions. Section A. Question number 1 to 12 are multiple choice questions. Only one of the choices is correct. Select and write the correct choice as well as the answer to these questions. Question 1. An approach that utilizes analysis of genome present in different environment to screen presence of variety of molecules. Answer A. Metagenomics. Question 2. In animal cell structure, the CO2 levels, that is carbon dioxide levels, in the incubator is usually maintained at answer C. 5 to 10%. Question 3. If we know where the genes are in each genome, it's difficult to count them due to answer B. 1 and 2. One is splice variants and second is overlapping genes. Question 4. Avocado growers in Kerala want to find a way to genetically engineer their crop to prevent it from ripening during shipping. This can be achieved by introducing answer C. Ethylene forming genes. Question 5. Microbial species used for commercial production of citric acid is answer B. Aspergillus niger. Question 6. To reduce the possibility of realization of vector without picking insert, the following method or methods may be used. Answer D. Second and fourth. Second is use of alkaline phosphatase and fourth is hybridoma technology. Question 7. In a comparative cDNA microarray, the cDNA obtained from the normal and diseased cells are labeled with red and green fluorescent dyes respectively. The yellow color of that spot indicates answer D. Gene expressed in equal measure in both types of cells. Question 8. Which disease has its locus on chromosome number 1 and 6? Answer B. Migraine. Question 9. A molecular disease which leads to increased hydrophobic interactions within the hemoglobin protein is answer B. Sickle cell anemia. Question 10. Interleukin 2 produced using CHO cell line is used for answer B. Cancer therapy. Question 11. Proteome of a given cell is dynamic because answer A. In response to internal and external changes, the biochemical machinery of the cell could be changed. Question 12. A novel gene that codes for the epitope has been introduced into yeast cells to produce answer A. Hepatitis B vaccine. Question number 13 to 16 consists of two statements, assertion A and reason R. Answer these questions selecting the appropriate option given below. Question 13. Assertion A. Patients who are administered monoclonal antibodies against CD3 can accept renal allograft. Reason R. Monoclonal antibodies block T cell function. So friends, answer is A. Both assertion A and reason R are the true and reason R is the correct explanation of assertion A. Question 14. Assertion A. Foaming is a problem in most microbiological cultures. Reason R. It is caused due to the presence of fatty acids and silicones in the culture medium. So friends, answer is C. Assertion A is true and reason R is false. Question 15. Assertion A. Yeast cells having YEP plasmid can grow on a medium lacking leucine and hence can be selected. Reason R. LEU2 gene codes for an enzyme required for the synthesis of amino acid leucine. Answer A. Both assertion and reason are true and the reason is the correct explanation of the assertion. Question 16. Assertion A. Long-term callus and cell suspension cultures and plants developed from such cultures may have chromosomal variations called somaclonal variations. Reason R. Somaclonal variations may lead to improvement in crops like disease resistance in potatoes. So friends, answer is B. Both assertion and reason are true, but reason is not the correct explanation of assertion. Section B. Question 17. The following table lists the common cloning vectors with the size of insert that can be cloned into them. Mark A, B, C and D in the table. A, B, C and D. Column 1 is vector type and column 2 insert size in KB. Answer: vector type plasmid, insert size 0.5 to 8. Bacteriophage lambda 9 to 23. Next cosmid 30 to 40. Next BAC 50 to 500. Next YAC 250 to 1000. Question 18. Based on microscopic observation, how can pathologists differentiate between cancerous and non-cancerous cells? Answer: cancerous cells, they do not exhibit contact inhibition. Non-cancerous cells, they exhibit contact inhibition. Cancerous cells, they pile on each other due to uncontrolled growth. Non-cancerous cells don't pile on each other. Cancerous cells more rounded in shape. Non-cancerous cells less rounded in shape. Question 19. Attempt either option A or B. A. What is inverted microscope and why is it used instead of compound microscope for observing animal cells in culture? Answer: in animal cell cultures, cells are at the bottom of the containers and hence can be visualized only by an inverted microscope in which the optical system is at the bottom with the light source on top. B. Why are B lymphocytes fused with myeloma cells in hybridoma technique? Give an important application of this technique. Answer: monoclonal antibodies are produced by fusing antigen activated B lymphocytes that have been immortalized with myeloma cells so that the hybrid cells retain the ability of B cells to secrete antibody and the ability of myeloma cells to grow indefinitely. Hybridoma technology has revolutionized the area of diagnostics and antibody based therapies. The availability of monoclonal antibodies has helped in early detection of many infectious diseases like hepatitis and AIDS. So friends, you can write any one of the following. Question 20. Why is 2D gel electrophoresis considered a suitable technique for studying proteomics? Answer: both IEF and SDS page are powerful techniques which separate proteins on the basis of isoelectric point and molecular weight respectively. Proteins are separated over a large surface area in two perpendicular directions. The resolution obtained is very high. Question 21. Which database was created to manage the redundancy in EST data? What is the role of the curator in bioinformatics? Answer: Unigene was created to manage redundancy in EST data. A curator is the one who checks the newly submitted data in bioinformatics for accuracy. Section C, question 22a. Why has sickle cell trait been selected in population where malaria is endemic? Answer: sickled RBCs resist malarial infection, hence safeguarded that population. B. What is the molecular basis of sickle cell anemia? Answer: the substitution of glutamic acid with valine in SCHB results in increase in hydrophobic interaction between the HB molecules, resulting in aggregation and ultimately leading to deformation of RBC to a sickle shape. C. How can we differentiate between a normal RBC and sickle cell RBC? Answer: shape of the sickle cell RBC is like that of sickle. Question 23. Why is the nutrient medium autoclaved before using it for culturing microbes? How would you sterilize a heat labile substance such as antibiotic solution? Answer: autoclaving is an important process in microbial cell culture. Autoclaving means heating the desired nutrient or equipment at 15 psi at 121 degrees Celsius for 15 to 20 minutes. The nutrient medium is autoclaved before using it for culturing microbes to destroy the microbes, fungal spores and bacteria present in the medium. To sterilize a heat labile substance such as an antibiotic solution, we make use of techniques like ultrafiltration. In this technique, we make use of membrane filters whose pore size is usually less than 0.5 mm. Question 24a. Identify A, B, C and D in the following table showing some industrially important plant secondary metabolites, their plant source and uses. So first column is product, second column plant source, third column uses. So friends, answer is first product vincristine, plant source Catharanthus roseus, uses anti-carcinogenic. Second product plant source Papaver sep, uses analgesic. So product B is codeine. Third product quinine, plant source Cinchona officinalis, uses anti-malarial. Next product Taxol, plant source Texas sep, uses anti-carcinogenic. B. The indiscriminate use of such valuable medicinal and other plants has brought them to near extinction. Suggest a possible solution to this. Answer. A possible solution is provided by cell and root cultures. Question 25. How does the charge relay system operate in chymotrypsin? Answer. A chymotrypsin folds bringing together ASP102, HIS57, SER195 in this sequence in space. ASP102, HIS57 and SER195 lie in this order forming a charge relay. The negatively charged aspartate carboxylate residue pulls the SEROH proton through HIS, leaving it with a negative charge. SER195 becomes acidic due to the unique constellation of the three amino acid residues because the protein has folded uniquely in space. Question 26. The gene for a eukaryotic polypeptide hormone was isolated, cloned and overexpressed in a bacterium. After the polypeptide was purified from the bacterium, it failed to function in the organism from which the gene was isolated. Suggest all the possibilities why the recombinant protein was inactive. Answer. To have proper three-dimensional folding, removal of introns is not there in the prokaryotes as they lack intron removal machinery, modification of proteins, that is post-translational modification, is not there. Question 27. Illustrate essential steps involved in raising a chimeric mouse using stem cell technology. Answer. Stem cells could be used to create chimeric mice by taking ES cells from a black mouse and implant it into the embryo of an albino mouse, that is white. The progeny so developed had skin color of black and white. Creation of chimeric mice using ES cells. Embryo donor black, ES cells, Trophobla st, culture of ES cell. Embryo host albino, micro injection of ES cells into embryo, inject embryos into uterus of surrogate mother. Chimeric progeny mice. Question 28. Attempt either option A or B. A. Selection is an important step in genetic engineering. You are given ampicillin and tetracycline antibiotics. Using these antibiotics, which selection technique could be used to differentiate between recombinant and non-recombinant cells? Answer. Replica plating. Host cells are first plated, master plate, on solid media with the desired antibiotic overnight. Velvet paper is aligned, pressed on master plate. With the same alignment, it is pressed onto the replica plate. Keep it overnight. Transformed colonies will now grow in replica plate. The colonies having insert can easily be scored off from master plate by comparing the two plates. Or B. Which microbial cells have been used extensively for functional expression of eukaryotic genes and why? Answer. Yeast cells have been used extensively for functional expression of eukaryotic genes because of several features. Yeasts are the simplest eukaryotic organisms, unicellular, and like E. coli have been extensively characterized genetically, easy to grow and manipulate, and large amounts of cloned genes or recombinant proteins can be obtained from yeast cultures grown in fermenters, that is large culture vessels. Section D, question 29. The below figure depicts a process used in diagnosis of a disease. A. Identify the mutation and the disease shown in the diagram. Answer. Reciprocal translocation and the disease is CML. B. Which technique can help in the identification of this technique? Answer FISH. Fish. Attempt either subpart C or D. C. Write down the steps occurring during NIC translation. Answer. The enzymes DNA polymerase 1 makes DNA and DNase 1 cuts DNA and are combined in a buffered reaction with DNIPs including DUTP labeled with a red or green fluorescence. Or D. How do extent of severity of the disease be detected in the above process? Answer. The status of the disease could easily be identified by counting the number of cells which appeared yellow. Further, it was possible to monitor the effect of chemotherapy and drugs by taking out samples and counting the number of cells appearing yellow. Question 30. Isolation of microbial product. Once the fermentation is complete, it is necessary to recover the desired metabolite. Minimally, this will involve separation of the cells from the fermentation broth. But it may also include purification of the metabolite with or without cell disruption. Cell disruption will be necessary if the metabolite is intracellular. Such operations are referred to as downstream processing. The steps involved in isolation of the desired microbial product are: first, separation of cells from the fermentation broth; second, cell disruption if the product is intracellular, or concentration of the both if the product is extracellular; third, initial purification of the metabolite; fourth, metabolite specific purification in which the metabolite of interest is purified to a high degree; and fifth, polishing of the metabolite, bringing it to 98 to 100% purity, where it is further concentrated and formulated for use. A. Cite an example of pure metabolite that can be obtained from above depicted flow chart. Answer. Streptomycin, any other relevant antibiotic. B. Suggest a suitable term which includes all the steps mentioned above. Answer. Downstream processing. Attempt either subpart C or D. C. In the second step mentioned above, which part will you use to isolate intracellular product? Answer. We will disrupt the cell and concentrate the product. D. Why is it generally recommended to have minimum number of steps in these isolation processes? Answer. Minimum the number of steps, more will be the purity and yield of the metabolite. Section E. Question 31. Attempt either option A or B. A. A protein with molecular weight of 10,000 contains 4, 3, 2 and 1 charge. Calculate the MZ ratio of protein ions at which mass spectrometer detects them. Draw the mass spectrum corresponding to the above mentioned ions. Answer. First, the mass spectrometer detects the protein ions at MZ 2501, 3334, 5001 and 10001 respectively. Friends, this is the diagram. Or B. A doctor has to prescribe a protein-rich diet to sportsmen to improve their performance. What are the three parameters that the doctor should consider while prescribing these protein sources? Answer. Essential amino acids and BCAA profile. Essential amino acids are those amino acids which have to be obtained from food and cannot be made in our cells. The branched chain amino acids BCAA are essential for the biosynthesis of muscle proteins. They help in increasing the bioavailability of high complex carbohydrates intake and are absorbed by muscle cells for anabolic muscle building activity. Biological value BV measures the amount of protein nitrogen that is retained by the body from a given amount of protein nitrogen that has been consumed. It has been observed that the BV of whey proteins is the highest compared to rice, wheat, soya and egg proteins. Protein efficiency ratio PER. PER is used as a measure of growth expressed in terms of weight gain of an adult by consuming 1 gram of food protein. The PER value of the following proteins are arranged in decreasing order: first whey, next milk, then casein, then soya, then rice and last wheat. Question 32. Attempt either option A or B. A. Write the basic steps with principles starting from an explant. Involved in obtaining a regenerated plant. Suggest any two applications of plant tissue culture. Answer. The basic technique of plant tissue culture involves the following steps. First, selection of suitable explants like shoot tip, leaf, cotyledon, and hypocotyls. Second, surface sterilization of the explants by disinfectants, example sodium hypochlorite, and then washing the explants with sterile distilled water. Third, inoculation, transfer of the explants onto the suitable nutrient medium, which is sterilized by autoclaving or filter sterilized to avoid microbial contamination in culture vessels under sterile conditions, that is in laminar flow cabinet. Fourth, growing the cultures in the growth chamber or plant tissue culture room having the appropriate physical conditions, that is artificial light 16H photo period, temperature 26 Celsius and relative humidity 50 to 60%. Fifth, regeneration of roots and shoots from cultured plant tissues and their elongation. Sixth, transfer to the greenhouse and then to fields. Application of plant cell culture, micropropagation, producing virus-free plants, producing artificial seeds, embryo rescue in interspecific or intergeneric hybrids, generating haploids or triploids, somatic hybrids and hybrids, in vitro germ plasm conservation, somaclonal variations, production of secondary metabolites. You can write any two applications. B. Indicate how intergeneric somatic hybrids can be made. Give an example. What are hybrids? How are edible vaccines advantageous over recombinant vaccines produced by bacterial fermentation? Answer. The protoplasts are isolated from two species of different plants and are allowed to fuse with each other in the presence of fusogenic agents like polyethylene glycol (PEG), most widely used and most successful method for protoplast fusion, or by electrofusion. The required fusion products, hybrid cells, are selected by various methods such as the use of different antibiotic markers or fluorescent dyes for two different protoplasts. Hybrids, cytoplasmic hybrids. Hybrids through protoplast fusion in which the genomes of one of the partners is lost. Edible vaccines offer following advantages over conventional vaccines: low cost, alleviation of storage problems, easy delivery system by feeding. You can write any other relevant point. Question 33. Attempt either option A or B. A. Who invented the di-deoxynucleotide chain termination method? Schematically explain the method. Answer. Dr. Frederick Sanger. Sanger's method. Whenever ddNTP comes in the DNA synthesis, further synthesis of DNA stops due to non-formation of 3,5-phosphodiester linkage as in ddNTP. There is 3H instead of 3OH. Structure of any ddNTP. Dideoxyribose is a pentose sugar with oxygen atom removed from each 2 and 3rd position. It must include the following reagents: single strand DNA which needs to be sequenced, a primer with a free 3OH, DNA polymerase, dNTPs, ddNTPs, 1% of total dNTPs. Method: primer extension in 4 different tubes, each containing a specific ddNTP at low concentration. Termination at the point where ddNTP is incorporated. Gel electrophoresis, autoradiography, positive reading of gel sequence. B. Why is dye termination method considered better than chains termination technique? Any two. Why is the autoradiogram read from bottom to top? Anode to cathode. Why do bacteria produce restriction enzymes and how do they protect their own DNA from its action? Answer. Dye termination method is automated, doesn't use radioactive isotopes so is safer, uses single lane agarose gels, fewer steps needed. An autoradiogram is read from bottom to top because for arriving at the original sequence from 3 to 5, every C is read as G, T as A as T and G as C as we arrive at the sequence from anode to cathode. Bacteria produce restriction enzymes to restrict the multiplication of phage genome. Bacteria protect their own DNA from phage action by methylation of its restriction site available in chromosomal DNA. Friends, we have completed sample question paper for biotechnology for class 12. If you like the video, kindly like it and share it with your friends. Thank you for watching.